Hi everyone. In continuation with my lecture series in pharmacology, today I will be talking on some of the aspects of pharmacokinetics. Pharmacokinetics is what the body does to the drug. It is the study of absorption, distribution, metabolism, and excretion of a drug. Now, pharmacokinetics suggests the choice of the drug, the route of administration, that is oral or injectables, etc. It also determines the dose of the drug and the dosing frequency. The pharmacokinetics determines how fast the drug is absorbed, how long it is going to stay at the site of action. That is, what is the onset of action and what is the duration of action. The foremost thing about the drug action is its absorption. Drug has to reach at the site of action after absorption by any of the routes which are administered. The drug absorption depends on the molecular structure, depends on the lipid solubility of the drug, high solubility, high absorption. It also depends on the degree of ionization of the molecule. For the drugs which are less lipid soluble, the ionization is an important determinant of the drug absorption. Highly ionized drug is poorly absorbed, and poorly ionized drug is easily absorbed. The drug absorption also depends on binding to tissue and serum protein of the drug, because the bound form of the drug is not available for drug absorption. The rate of absorption depends on the route of administration, if it is oral or injectable. It also depends on the drug formulation, whether it is oral dosage form, solid dosage form, or it is a liquid dosage form. If it is injection, whether it is an aqueous injection or it is a depot injection, etc. The extent of absorption, that is the total amount of absorption, depends on the lipid solubility and also presence of efflux mechanism by PGP. PGP is a protein which efflux the drug out of the cell. Drug absorption essentially involves two processes. One is passive transport, that includes paracellular transport as well as simple diffusion. The active transport may be either facilitated diffusion or through drug transporter mechanism requiring energy. The passive diffusion, in this the drug diffuses along a concentration gradient due to its lipid solubility. Higher the solubility, better is the diffusion. For poorly lipid soluble compounds, which are ionic, the passive diffusion depends on the electrochemical gradient and the pH across the membrane. Acidic drugs will be absorbed more in the acidic medium because they are non-polar, and basic drugs in alkaline medium because they are non-polar in this alkaline medium. So we see that acidic drug, say for example aspirin, which is an acidic drug, will be absorbed more in the gastric pH than the small intestine, and so is true for basic drug. The absorption also depends, or the passive diffusion also depends on the surface area of the membrane. In the GI tract, the small intestine has largest surface area, and so the most of the drugs are absorbed from the small intestine. Based on the similar principle, we can say that alkaline urine causes increased excretion of aspirin and urates, because in alkaline urine the acidic substances will be more ionized and so available for excretion. Some of the antihistaminic second generation or even third generation antihistamines are ionized molecule, So they are poorly lipid soluble and not available to enter into the brain. So they do not penetrate the brain easily, do not cause the side effect of drowsiness. The active transport is by two mechanisms. One is facilitated diffusion involving carrier proteins in the direction of electrochemical gradient. Say for example, a transport protein or the carrier protein OCT1, it facilitates the movement of thiamine and metformin. The active transport, which is against the electrochemical gradients, it requires energy. Say for example, PGP for steroid hormone, sodium potassium ATPase for digoxin, and PGP for so many other drugs. Coming to bioavailability, bioavailability is defined as the fraction of unchanged drug reaching systemic circulation by any route. So it is unchanged drug, that is important. For intravenous drug, bioavailability is assumed to be 100% because intravenous drug make the drug available into the bloodstream almost 100% without any metabolism, without any interference of absorption. Whereas oral drug has bioavailability which is usually less than 100% due to incomplete absorption or first pass effect. There are several factors which affect the bioavailability of the drug. The important one is pharmaceutical factor. Pharmaceutical factor means could be what is the dosage form? Is it a solid dosage form or liquid dosage form? If it is a solid dosage form, it depends on the dispersion, the dissolution profile of the tablet or the capsule, how fast it is dissolved, and also on the disintegration of the tablet. And there is a very well-known incidence of phenitoin toxicity that happened in 1968 because of the change of excipient. The change of excipient from the original, from the initial process to a lactose. It was changed from some excipient to lactose as one of the excipient, which increased the bioavailability of the drug, and so there were a lot of incidences of phenitoin toxicity, that is ataxia. Similarly, digoxin has got a very variable response sometime due to the change in the manufacturing process. So the bioavailability also depends on the dosage form, it depends on the use of excipients, and it also depends on the manufacturing process. The bioavailability depends on the fasting condition or fed condition. There are certain drugs which are better absorbed in the fasting condition, whereas certain drugs which are better absorbed in the fed condition. So this is also very important while administering the drug to the patient. Then disease of gut and other organs like liver, kidney also affects the availability of drug into the body. Drug metabolism is an important determinant of bioavailability. If the drug is highly metabolized, quickly metabolized, the bioavailability will be less, and vice versa. Then drug efflux through BGP or ABCB1. These are the proteins which take the drug out of the cell. If that mechanism or drug efflux mechanism is active for a particular drug, the bioavailability will be poor. And last but not the least is the route of administration. The drug which is given by injections has got a better bioavailability than a drug which is given by oral or by any other route. We'll discuss this route of administration in subsequent slides. Just to have a glance on the bioavailability from different routes, see intravenous almost 100% or 100%, and that is why it has got the rapid onset of action. The intramuscular route has the bioavailability of 75 to 100%. Subcutaneous route, which is used below the skin, the bioavailability is 75 to 100%. The oral route has the bioavailability of as low as 5% to as high as 100%. It is a very convenient route. And the low bioavailability due to oral is because of first pass effect, we'll discuss this point later, or extensive metabolism by any other, or poor absorption. The rectal administration, some of the drugs are used by rectal route, and in this case the bioavailability is 30% to as high as 100%. In rectal administration also, the drug escapes metabolism partially. Inhalation, the drug which is taken by this route shows bioavailability of as low as 5% to as high as 100%, and usually they are having a rapid onset of action. The transdermal route, the drug which is applied on the skin, they have the bioavailability of 80 to 100%, but it is a slow absorption. Just to have a basic idea about two different types of formulations which are used by oral route. One is IR, which is known as immediate release. IR is formulation has got a quick onset of action, it has got a fast excretion or fast elimination, duration of action is short, and then it requires a frequent dosing. They are to be given twice, thrice, or even 4 times a day. Whereas sustained release formulation, which are manufactured, which are developed by changes in the excipients and certain technology by the pharmaceuticals, they are slow release form of the drug, they have a uniform absorption, they have a prolonged action, they require less dosing frequency, and they have a lesser side effect because of their low Cmax. The example is carbamazepine. And another advantage of sustained release formulation is the better patient compliance due to less frequent dosing. Most of the tablets are required to be taken once in a day or max twice in a day. The limitation of sustained release formulation is erratic absorption, overdose due to dumping, and it cannot be divided, and usually sustained release are slightly more expensive than IR formulation. Coming to the next part after absorption is distribution. After the drug is absorbed into the bloodstream, it goes to the various tissues of the body like liver, kidney, bones, fats, and so on and so forth. The distribution depends on the lipid solubility, state of ionization, protein binding, regional blood flow, and cardiac output. All these factors they determine that to what extent the drug will be going to a particular tissue. The lipid insoluble drugs are confined to plasma and interstitial fluid like gentamicin and streptomycin because they do not cross the plasma membrane. So most of the lipid insoluble drugs, they remain in plasma and interstitial fluid. They are not going to the tissues. Whereas highly lipid soluble drug, they first enter the brain, heart, liver, followed by fat and the muscles. The volume of distribution is calculated based on the doses and the plasma concentration, which is volume of distribution is directly proportional to the dose administered, and it is inversely proportional to the plasma concentration. Greater the lipid solubility, faster is the redistribution. One good example is thiopental sodium, which is an injectable anesthetic drug used for sedation in the pre-anesthetic phase. Now this drug is highly lipid soluble. The moment it is injected intravenously, it immediately enters to the brain within few seconds. The patient feels drowsiness, but the drug leaves the brain as fast as it goes in, and it goes to the fat tissues. So the onset of action of this drug is very, very short. The patient is awakened only after a couple of minutes, but the drug remains in the body for a longer time because it is redistributed into the fat. Similarly, diazepam, which is also a lipid soluble drug, has got a short action of 6 to 8 hours in spite of long elimination half-life of more than 30 hours. This is again because of its high lipid solubility. Then distribution depends on the protein binding, plasma protein binding. If there is a high protein binding, The tissue distribution is poor because it cannot enter into the tissues because of lipid solubility. There are certain drug interactions which we are considering worth mentioning, like aspirin and sulfonylurea. Sulfonylurea is a drug which is used for the treatment of diabetes. Now, aspirin and sulfonylureas both they bind to the same site in the plasma protein, and aspirin has got a higher affinity, so it displaces the antidiabetic drug from the site, and so the concentration of antidiabetic drug increases, leading to hypoglycemia. So it is advised not to give aspirin and sulfonylurea, or if they are given together, they should be used with caution. Similar interaction is seen with vitamin K and bilirubin, where vitamin K has got high affinity for protein than bilirubin, and so it displaces the bilirubin from the bind site, leaving free bilirubin entering into the brain, causing kernicterus in infants. Then the tissue storage, large volume of distribution, long persistence in the body may have a long duration of action. Say for example, chloroquine is stored in the liver, kidney and retina. Thiopentol in the body fats, as we have already discussed. Iodine is stored into the thyroid gland. Tetracyclines, heavy metals like lead and others, they are deposited or they are stored into the bone and the teeth, which can cause various side effects. Aminoglycosides like streptomycin and gentamicin, they are stored into the kidney and ear, leading to toxicity like kidney toxicity or nephrotoxicity and ototoxicity. So storage into the tissues of various drugs may lead to toxicity, or it could be a concern for a particular drug. Then in the distribution we see that there is the drug usually do not cross the brain because there is a barrier which is composed of capillary endothelial cells and the glial cells surrounding the pericapillary membrane. And this blood-brain barrier does not allow all the substances or all the drugs to pass so freely. And moreover, there are tight junctions in the brain capillaries which does not allow the drug to move freely or only allows the transcellular transport. So we can say that blood-brain barrier restrict the entry of drug into the brain. One of the important aspects of drug to enter the brain is lipid solubility. Say for example, we have seen thiopentol sodium, diazepam and so many other drugs. Highly lipid soluble drug is more accessible to the brain. However, this blood-brain barrier may have some modification, some alteration in conditions of meningitis or encephalitis and may allow certain drugs to enter. For example, the antibiotics which are otherwise not crossing the blood-brain barrier can cross the blood-brain barrier to a therapeutic level in patients with meningitis and encephalitis. Similarly, there is a placental barrier which protects the fetus during pregnancy, and the deciding factor for the drug to cross the placental barrier is lipid solubility, protein binding and ionization. And there is an efflux of drug from placenta to the maternal circulation. So there is a mechanism which restricts the use of drug to the fetus. However, it is considered that to some extent all the drugs they will reach to the fetus if taken by mother during pregnancy. So based on that, the drugs are categorized into various safety categories A, B, C, X, etc. on the basis of safety to the fetus. For example, category X drug is considered to be absolute contraindication because there is a teratogenic effect or that may cause congenital malformations. Thank you.